So children, in the last part you had studied about the post-fertilization events. So in that we had studied about how the embryo is formed. We started with the double fertilization and the different stages of embryo development. We studied how what is the pro-embryo stage, two-cell stage, apical cell and basal cell. Then the globular embryo, which leads to the formation of the heart-shaped embryo where the cotyledon starts appearing, and finally the mature embryo, which has a radicle and the plumule with the meristematic tissues are there present in it, which divides very frequently. And then we have studied about the cotyledons, that is the structure of a mature embryo of a dicot plant. Now let's study a little bit more detail of what is the structure of dicotyledonous embryo over here, and this is the structure of monocotyledonous embryo. So let's study these two in detail. Now, when the embryo develops, that is, that angiosperms, we can classify that based on the number of cotyledons present into dicot or monocot. In dicots, it has two cotyledons, and in monocots, the plants, that is rice, wheat, maize, has one cotyledon in it. So that's how we can classify the angiosperm, that is a flowering plant. Here the dicot is nothing but the mango, all that comes under the dicot plant. Now in the structure of a dicotyledonous embryo, very important. The main structure is nothing but it has a embryonal axis. This is the axis over here, and the important part is above the embryonal axis you have the part known as epicotyle. Above the embryonal axis, here in the center, I told you we had studied in the previous one. This is the plumule, which is nothing but consists of lot of meristematic tissues which give rise to the shoot system with the meristematic tissues. Just after that is between this plumule and the embryonal axis, you have the cell, that is a structure known as epicotyle. Epicotyle growth and all, it will help the growth of the epicotyle. It will be helpful during the germination of the seed. Whether the seed decides whether the seed will germinate, the germination is epigeal germination or hypogeal germination. That you will be studying in the later part of the chapter. Now, this part here, these are nothing but the cotyledons over here, and this structure in between the two cotyledons. It's a dicot, so these are the cotyledons, and in between the cotyledons there are many tissues present over here. This is nothing but the plumule which gives rise to the shoot system. Alright, and in between that is the embryonal axis. So above the embryonal axis, this part is known as epicotyle. Below the embryonal axis, this part is known as hypocotyle, and this hypocotyle, it will be extended towards the lower end of the embryo, and this whole structure, this is known as radicle. Radicle is nothing but it gives rise to the root system. So when the seed germinates, after germination, when it is growing, so it will have the shoot system and the root system. Root system grows under the ground, shoot system grows above the ground. So in the shoot system, you have the epicotyle and the plumule. This is a embryonal axis. Below the embryonal axis, you have the hypocotyle. Like the epicotyle, hypocotyle growth also will tell what kind of germination takes place in the seed, whether it is epigeal germination or hypogeal germination. Then you have the radicle which gives rise to the root system, and the radicle is protected by certain cells over here. We call it as root cap. It is found in mango and all that. So it gives a protection to the root, the primary root, and then the secondary root and tertiary roots will develop from that. So this is the structure of a dicotyledonous embryo. So what are the main parts? So please draw the diagram in your notebook neatly and then label the part, that is very important. So this is the half you can do. Suppose this is the two cotyledons over here, and this is the shoot system, this is the root system over here. So this is the axis. So this part is the epicotyle, that is embryonal axis. Thus above is epicotyle, this is the plumule, and below the embryonal axis this will be the hypocotyle, and and then below that is a radicle. So epicotyle, plumule is this in the middle between two cotyledons. These are the two cotyledons, that is a dicot. Then you have the hypocotyle, and then hypocotyle extends to radicle, and radicle is covered by the root cap. Now let's study what is the structure of the monocotyledonous embryo. That is, what is the ploidy of that? It is also, these both are diploid in nature, two sets of chromosome. Okay, now it is quite different, so please you can analyze it comparatively. So here, if you see, this is the embryonal axis which divides the plant, that is above the shoot system, below the root system is formed. So that is meant by the embryonal axis. So in case of monocotyledonous embryo, this is the embryonal axis. The cotyledon, there is only single cotyledon. Cotyledon grows on the lateral side of the plumule and all. It grows on the lateral side. This is the cotyledon. The name of the cotyledon for monocot is nothing but scutellum. Scutellum is nothing but cotyledon. What is the purpose of cotyledon? Cotyledons will provide nourishment. It has lot of supply of food in it. Minerals and all are there, lipids are there, proteins and all. So it gives the nourishment to the growing embryo. So that is the main purpose. So the cotyledon of a monocot seed or embryo is known as scutellum. It is found as a lateral shield-like growth on the side. This is the embryonal axis. Now as I told you, embryonal axis, below the embryonal axis you have, this part is nothing but this part below the embryonal axis. This is the radicle. Radicle extends into root cap. Root cap protects the radicle. So you have the below the embryonal axis you have radicle, root cap, and root cap is covered by a sheath like a cover structure, and that covering structure is known as coleorhiza. So please understand it properly that the coleorhiza main purpose is it is found below in the maize and all. Maize, wheat, rice, they all are monocot. So below that it has the structure, sheath like structure known as coleorhiza, and this coleorhiza, when the seeds germinate, it remains under the soil. It is non-green in color. Now you come to the upper part. There is no hypocotyle or epicotyle, so the parts below the embryonal axis is radicle, root cap, coleorhiza covers the radicle below the embryonal axis. Now look at the parts above the embryonal axis. So above the embryonal axis you have this structure over here. This is nothing but which is covered by a leaf primordia. That is the baby leaf which grows, which can synthesize food during that is when it comes out of the soil. It starts the photosynthesis. So this middle part which is covered by, which has lots of meristematic tissue, this is the shoot apex. And this on the side, it is the epiblast. All the structure, it gives a nourishment to the embryo. And then you can see here, this whole thing is nothing but the cotyledon there. And then not the cotyledon, it is known as coleoptile. So what is coleoptile? Coleoptile covers the shoot apex. Shoot apex is nothing but it gives to the shoot system, and then this part, coleoptile, will come out of the soil during the germination, whereas coleorhiza will remain under the soil during the germination of the seed. And the cotyledon of a monocot plant is known as scutellum. So it is found on the lateral side, it is not along with the structure of the other part. So this is the structure of a mature, that is monocotyledonous embryo. Very important part, so please learn it. This is also to be asked for the 3-mark diagrammatic question, and you have to label the parts. Identify the part. What is this part known as? What is the function? Coleoptile, it covers the shoot apex. Okay, and then you have, this is the embryonal axis. Below the embryonal axis there is radicle. Radicle gives rise to the root system, which is in turn covered by the root cap protection, so that because it has to grow under the soil, and that is in between the rock and all, hard surface and all. So root cap just gives a protection to radicle, and this whole thing is covered by a sheath-like structure which is known as coleorhiza. So these are the parts. Please see the comparative analysis, draw the diagram neatly and label it and learn. So I will be just, you can see the slides and you can understand the whole structure once again. I hope you have understood this well. Please label and then write the notes and submit it. So children, let's summarize what we have learned today. So first we had studied about double fertilization followed by triple fusion, then the formation of endosperm, then development of embryo. Two types of embryos we had learned. So first we'll see the structure of monocotyledonous embryo, then the structure of dicotyledonous embryo. Now look at the slides. So we started with the first one. So stage, that is of fertilization. Once the pollen tube reaches the synergid, it releases two male gametes into the cytoplasm of a synergid. One of the male gamete fuses with the egg nucleus to form a diploid cell known as zygote. The ploidy of the male gamete is haploid and the ploidy of the egg cell is also haploid. So, male gamete fuses with the egg cell to form zygote, which is diploid in nature, and this process is known as syngamy. The other male gamete moves towards the center, and it fuses with the polar nuclei in the central cell. There are two polar nuclei, so just before fertilization, two polar nuclei fuses to form the secondary nucleus. So the ploidy of that is diploid. So the other male gamete fuses with the secondary nucleus or the polar nuclei to form primary endosperm nucleus. So the ploidy of that will be triploid, 3N. So that process is known as triple fusion. Both the syngamy and the triple fusion occur simultaneously. So this process is known as double fertilization, which is an important characteristic features of all angiosperms, flowering plants. Then, soon after this process, the synergid starts degenerating at the micropylar end and the antipodals will start degenerating at the chelazal end. The function of the synergid is also to provide the nourishment to the egg cells and all. Then the central cell gets converted, that is it undergoes division and then it forms primary endosperm cell or PEC, and this is primary endosperm nucleus which is triploid in nature and it is known as PEN. The zygote later develops into embryo and the primary endosperm nucleus later develops into the endosperm. Endosperm, that is, which gives nourishment to the developing embryo. Now this is again double fertilization what we had studied. So here it is the zygote which is formed, that is a male gamete fusing with the egg cell to form zygote which is diploid in nature, and in the central cell you can see three cells over here, that is two of the polar nuclei and one of the male gamete, resulting in a triploid endosperm or primary endosperm nucleus. So it is a complex mechanism of fertilization in flowering plants, angiosperms especially, and it is the joining of female gametophyte with the two male gametes. One sperm nucleus unites with the egg to form zygote, whereas the other fuses with the two polar nuclei to form endosperm, that is which provides nourishment to the growing embryo. So this whole process is known as double fertilization. So what we had learned, one of the male gamete fuses with the egg nucleus to form a diploid cell called zygote. This process is called syngamy. So the ploidy you can calculate it like that. Male gamete haploid, egg cell is also haploid. So these two will fuse to give a diploid zygote which later develops into embryo. In triple fusion, the second male gamete fuses with the two polar nuclei or the secondary nucleus at the center to produce a triploid primary endosperm nucleus. Two polar nuclei, each of them which are haploid, it fuses with a male gamete which is a haploid and it gives rise to triploid primary endosperm nucleus or PEN, and this process is known as triple fusion. So syngamy and triple fusion occur simultaneously, so this process is known as double fertilization. So now there are some questions which will be asked on this, how you have to calculate the number of chromosomes in the endosperm. So for that, as I told you before, when you look into the embryo sac, all the cells are haploid in nature except the polar nuclei which fuses to form diploid secondary nucleus. The antipodals are haploid in nature, synergids are haploid in nature, and the egg cells are haploid in nature. That means if there are 24 chromosomes present in the plant like monocot plant like rice, the haploid number of chromosomes in each will be half of that. That will be 12. So keep that in your mind and you have to do the calculation. There'll be many questions which will be asked on this basis. You have to find out the haploidy, that is the ploidy of endosperm or the antipodals or the egg cell or the zygote, etc. So for example, one of the question here, the myocyte of rice has 24 chromosomes. How many chromosomes are present in its endosperm? So you know the myocyte of rice which is given, that is 24 chromosome. So therefore the number of chromosomes in the, that is male gamete, will be haploid because it is formed by meiotic or reduction division. So it will have 12 chromosomes. Now the number of chromosomes in two polar nuclei, it will be 12 plus 12 which will be equal to 24. Therefore the number of chromosomes in its endosperm you can calculate is 24 plus 12, that is 36 chromosome. Now can you tell me what will be the ploidy of antipodals? The ploidy of antipodals will be, that is haploid, that will be also equal to 12 chromosomes. What will be the ploidy of the zygote? Zygote is diploid, so zygote will be having 24 chromosomes. So that's how you have to do the calculations when the questions are asked in this order. So now let's see what are the post-fertilization events. In post-fertilization events, there is development of endosperm from primary endosperm nucleus, the development of embryo from the zygote, then the development of seed from the ovule, and finally the development of fruit from the ovary. So we are going to study the first two events in this. We have studied the first two events in this class, that is development of endosperm and development of embryo. Now look at this diagram. This is the first step, development of endosperm from primary endosperm nucleus, which is triploid in nature. So this is the oospore, or that is a zygote, and this is the space for the endosperm present over here. So first the endosperm undergoes, look at this diagram, it undergoes free nuclear division. That means there is no cytokinesis, no cell wall formation. So it divides mitotically to form thousands of endosperm nuclei, and then they don't form any wall. So it is known as free endosperm nucleus, or free nuclear endosperm. After this, what happened? The cell wall starts developing. So this stage of endosperm development is known as the cellular endosperm. So in endosperm development, there are two stages. One is known as free nuclear endosperm and second one is known as cellular endosperm. And then the endosperm, the oospore divisions will also take place simultaneously to form the embryo, and these are the cellular embryo which you have seen it. So the cellular endosperm, that is a primary endosperm, first divides into two and finally divides into 8. So all are mitotic division and it leads to the formation of the cellular endosperm. So cell wall formation occurs immediately after division. Subsequent division also is accomplished by cell plate formation. As a result, endosperm becomes cellular from the beginning. And here this endosperm mainly, this development of endosperm happens before the development of embryo because endosperm provides nourishment to the developing embryo. That is why first this happens, it happens prior to the embryo development, and it occurs in many of all the dicot plants as well as in the monocot plants. Now this is the image to show you how the endosperm with the embryo and with the nucleus looks inside an ovule. This is the outer integument which we have studied, inner integument, and this is the nucleus. Nucleus, what is the ploidy? Nucleus is also diploid in nature. And this is the cellular endosperm over here. And what is the ploidy of that? That is triploid, 3N. And this is the embryo which is diploid and present at the micropylar end. This is a chalazal end. And these are the integuments, outer integument and inner integument. So this is how the ovule looks, that is after the fertilization. This is an example of a seed which shows the endosperm which provides food for the developing embryo or the plant. And another feature you can see, this is an example of the coconut, tender coconut. Tender coconut, it has the outer portion, that is a green color which you see, outer part of the coconut, it is nothing but exocarp. Then you have the fibrous husk which is in the middle, it forms a mesocarp, and the hard shell forms the endocarp. Then you can see the white kernel. White kernel is nothing but endosperm or meat. Endosperm which we eat. All right. And then you also have seen the water inside. The water inside the coconut is nothing but the free nuclear endosperm. So the white kernel is a cellular endosperm and the water is a free nuclear endosperm. Next. What is the function of the endosperm? It is the most nutritive tissue for the developing embryo in the angiosperm, and in gymnosperms like pinus and or pine cone, the female it represents the female gametophyte, whereas the female gametophyte in angiosperm differentiates before fertilization and is haploid. The endosperm is a product of fertilization and is usually triploid. After double fertilization, what is the egg called? It is called the zygote. And the fusion products of polar nuclei and the second male gamete is termed as the primary endosperm nucleus. Then this process is seen in all the angiosperm, but it does not, some angiosperm does not form endosperm. They are the families of Orchidaceae, that is orchids. They don't form the endosperm. Then Podostemacea and Trapeaceae. These both are aquatic plants or phytic plants, which will not produce the endosperm at all. Next, the second stage, we'll see how the embryos are developed in a dicot plant. So this is the zygote over here. So the zygote in the first stage, it will undergo asymmetric mitotic division. Zygote is diploid in nature to when it undergoes asymmetric, means it will form a large basal cell and one apical cell. The basal cell is this, it will be dividing, it will undergo the transverse division first, then the vertical division, forming a series of cells, a group of 6 to 10 cells, and this is called suspensor. Okay, and the first cell of the suspensor towards the this is a micropylar end, it is known as haustorium, whereas the last cell of the suspensor towards the chalazal end is called hypophysis. This hypophysis later develops into radicle which later develops into the root system. And then after this, the smaller, that is this is the basal cell and this is the apical cell. So this basal cell, it has formed the suspensor and all. Later the apical cell undergoes one vertical division and later it undergoes transverse division, and first it will form 4 embryonal cell, or it is known as quadrant stage here. And then later this again divides to form 8-celled embryo or octet stage. In the octet stage, the 4 cells at the apex give rise to plumule. That means it will form it will form the plumule which gives rise to the future shoot system, and the other 4 cells gives rise to the hypocotyle. That is hypocotyle followed by the radicle which forms the root system. Now, so here this is a suspensor, and then there are certain stages. Here when this is the pro-embryo stage where it is 2-celled. Here it forms like a spherical, so we call this stage as globular embryo. Later globular embryo starts dividing and it forms heart-shaped embryo. This is heart-shaped embryo stage. And then here you can see the cotyledon dicot. These two are the cotyledons which have started developing. And this in the center, it is having lots of meristematic, fast dividing, repeatedly dividing cells, meristematic cells, and this is the plumule. Then you can see this part, this is the radicle and which gives rise to the root system. This is the plumule over here which has lot of meristematic tissues, and these are the 2 cotyledons. And this stage is known as torpedo stage. So this is what we have seen, that is the angiosperm flowering plant, and this is the ovule over here, and the nucleus, endosperm nucleus in the middle, integuments, and then how the zygote, which is at the micropylar end, forms a terminal cell and the basal cell. Again it undergoes transverse and vertical division to form the proembryo. Proembryo forms a suspensor. This is the heart-shaped stage. Okay. So here it is a procambium, that is the different cells, how it is dividing repeatedly. And then this is the heart-shaped embryo having 2 cotyledons, plumule and the radicle, and this is the suspensor. And this is the whole structure, how it develops inside the ovule. Next, in the end, we are going to study what is the structure of dicot embryo and monocot embryo. This is the structure of a dicot embryo. The dicot embryo, it is the Here you can see imaginary line. Just think this is as an embryonal axis. It has embryonal axis and cotyledon. So this is like a dicot embryo, for example, beans, peas and all that. So this part is nothing but it is known as a cotyledon. The part between the cotyledon and this embryonal axis, here we have the epicotyle. Okay, and the middle part here, that is between the two cotyledons, this part is where lot of meristematic tissues and leaf primordia will be present. This is the plumule, which later develops into the shoot system. And these are the two cotyledon. Cotyledon, what is the function? It provides nourishment to the growing embryo. And here, if this is the embryonal axis, the part below the embryonal axis is known as hypocotyle. And hypocotyle is followed by the lower part, that is a radicle, and radicle is covered in few plants with a root cap. Now this whole radicle and the root cap gives rise to the root system. So the parts of a dicot embryo, you have the cotyledon, plumule, and this part is nothing but it is known as the epicotyle. Then below the embryonal axis there is hypocotyle, radicle and root cap. So the diagram will be given to you and you have to identify the parts and state the functions of each. So please draw the diagram neatly and label it. Another one, this is the monocot embryo like maize, rice, wheat. So there is the structure, there is slightly, it is different. So monocot, as the name suggests, it has one cotyledon. The cotyledon in a monocot is known as scutellum. And if this is the imaginary embryonal axis, the lower part in the embryonal axis, this is the hypocotyle region and this is the epicotyle, that is in the case of we have seen in dicot. So here this is the lower, that is below the embryonal axis, we have the radicle. Radicle is covered by the root cap, and below the root cap the entire radicle root cap is covered by a sheath like structure known as coleorhiza. Coleorhiza is found only in the monocot embryo. And then look at the upper region, that is above the embryonal axis. You have the epiblast over here, and then this is the shoot apex, this middle part, that is the part which gives rise to the leaves and all that. It forms the shoot, that is the shoot apex, and this shoot apex is covered by another sheath on top. It is known as coleoptile. Very important diagrammatic question, function will be asked. So the cotyledon in a monocot embryo is known as scutellum. The shoot is covered by coleoptile. This is a shoot apex, and then below the embryonal axis you have the radicle, which is followed by root cap, and it is covered by the coleorhiza. So this is the structure of dicot embryo and monocot embryo. Hope all of you have understood this. I want all of you to write the notes and draw all the diagrams neatly and solve the problems based on the ploidy of endosperm or antipodals. So thank you, children.